Aging is one of the most fundamental biological processes, yet its underlying cause remains a subject of intense scientific debate. Is it the result of a built-in biological program, a kind of self-destruct sequence, or is it the consequence of our bodies slowly accumulating damage over a lifetime? In this explainer, we will explore the evidence for these two competing theories.
So, to frame the central question, is there a clock ticking down inside of us, a preset program designed for decline? Or is aging more analogous to a complex machine that gradually breaks down from a lifetime of small accumulated errors that our repair systems can't completely fix? These two viewpoints lead to very different approaches for understanding and potentially intervening in the aging process. Let's first examine the case for programmed aging. For quite some time, this idea faced significant scientific challenges, but recent discoveries have brought it back into the spotlight. The primary reason for this resurgence is a remarkable tool known as the epigenetic clock.
Now, these aren't mechanical clocks. They're molecular. Epigenetic clocks function by measuring changes to our DNA, specifically a process called DNA methylation. You can think of methylation as a series of chemical tags that attach to our DNA, controlling which genes are active. It turns out that the patterns of these tags change in a very consistent, predictable way as we get older. This allows researchers to analyze a tissue sample and determine its biological age with a high degree of precision. To give you a sense of the scale we're dealing with, the human genome contains approximately 28 million of these potential methylation sites. The clocks don't need to analyze all of them. They function by measuring the patterns across a select subset of these sites to derive a biological age. And this is the core of the argument for programmed aging. The fact that these clocks are so accurate, predicting age from before birth all the way to over 100 years old, seems to suggest a deterministic process. For proponents of this theory, such precision implies that aging is not just a random series of events. It points towards a deliberate underlying biological program. So this brings us to the two competing frameworks. On one side, programmed aging, an evolved, intentional biological process. On the other, stochastic aging, an accidental byproduct of accumulated damage and imperfect repair. Now, let's turn to the alternative perspective. This view draws not from the idea of a program, but from fundamental principles of evolution and molecular biology. It posits that aging is not an intentional process, but rather the outcome of a continuous struggle against decay. This theory is grounded in evolutionary biology. You see, the force of natural selection is strongest up to the point of reproduction. It optimizes our bodies to survive and pass on our genes. But after we've reproduced, that selective pressure diminishes. This period is often called the selection shadow. In this shadow, there's less evolutionary advantage in investing energy into perfect long-term maintenance. The priority, from a genetic standpoint, has already been met. This evolutionary logic is paired with a fundamental principle of biology. The default state of complex systems is decay. Our molecules, like DNA and proteins, are not inherently stable. They require a constant input of energy for maintenance and repair. From this viewpoint, life doesn't require a special program for death. The challenge is the complex program required simply to sustain life. And this isn't just a theoretical concept. At the molecular level, our cells are under constant assault. We're talking about things like DNA double-strand breaks, damage from oxidation, errors in epigenetic marks, and misfolded proteins. Our bodies do have sophisticated repair systems to deal with this, but they aren't flawless. Over a lifetime, the unrepaired damage gradually accumulates. Experimental evidence provides support for this idea of randomness. For instance, studies on the nematode worm C. elegans show something quite telling. Even when you have populations of worms that are genetically identical clones and you raise them in precisely controlled environments, their individual lifespans can vary by a factor of 3. If aging were a strictly determined program, you would expect a much more uniform lifespan. This variability strongly suggests the influence of random stochastic factors. This brings us to a potential contradiction. If aging is fundamentally a random process of damage accumulation, how can these epigenetic clocks measure it so precisely? To resolve this, we need to reconsider what the clocks are actually measuring. The central argument here is that the clocks aren't tracking a program at all. Instead, they are measuring the predictable statistical consequences of accumulating random damage. A useful analogy is to think of them not as a countdown timer, but as an odometer on a car, which measures cumulative wear and tear. So,
How can randomness yield a predictable result? Well, let's break it down. In a young organism, the methylation patterns at a given location are relatively uniform across its cells. Over time, random errors, or epimutations, occur in individual cells. Any single one of these errors is unpredictable. However, when you look at the average change across millions or billions of cells, a clear, predictable trend emerges. This is a statistical principle. The clock is simply measuring this aggregate trend, which is remarkably consistent despite the randomness at the level of individual cells. We can see evidence for this trade-off between maintenance and reproduction when we compare different types of cells in the body. Our germline cells, the sperm and egg that pass on genetic information, are maintained with very high fidelity, showing a low mutation rate. In contrast, our somatic cells, which make up the rest of our body, have a mutation rate that is at least an order of magnitude higher. This points to an evolutionary strategy. Invest heavily in protecting the genetic blueprint for the next generation, while providing only good enough maintenance for the parent organism. This leads to a rather profound conclusion, which is summarized well by this quote. One does not need a program to die, only a program to live. From this perspective, our genetic code is not a program for death. It is an incredibly complex program for life, for building a body and maintaining it against the relentless forces of decay. Aging, then, is the eventual, unavoidable consequence of that maintenance program becoming overwhelmed.
So, if we accept this view, what are the implications? Understanding aging as a consequence of accumulating damage rather than a fixed program has significant consequences for how we might approach interventions. This framework shifts the goal of longevity research. Instead of searching for a single aging program to deactivate, the focus moves toward enhancing our body's own inherent maintenance and repair mechanisms. This means developing strategies to improve DNA repair, to more effectively clear out damaged proteins in cells, and to support the function of our cellular organelles. And we have compelling evidence that biological age is, to some extent, malleable. For example, during early development, the epigenetic slate is essentially wiped clean, resetting the clock to 0. We also see this in nature, where some organisms can enter states of suspended animation, which halts or even reverses aging. And in laboratory settings, cellular reprogramming using specific factors has been shown to reverse epigenetic age. These examples demonstrate that the accumulation of damage is not necessarily irreversible. This entire process can be framed in the language of information theory. You can think of aging as a form of biological information entropy. Over a lifetime, the precise information stored in our genome and epigenome is gradually corrupted by the accumulation of random errors and molecular noise. This progressive loss of information leads to a decline in regulatory precision, which manifests as the functional decline we call aging. This brings us to the final, forward-looking question. If we view aging not as a program, but as a battle against accumulating chaos, the challenge becomes, how can we bolster our innate biological defenses? The future of research in this field may not lie in searching for a kill switch, but rather in understanding how to reinforce and support the complex systems that work tirelessly to maintain order within our bodies.